Hello and welcome to Preprints in Motion. Join us as we sit down with early career researchers and discuss their latest preprint and find out about their journey through the muddy marshes of academia. But we don't stop there. Every month we'll be bringing you special episodes with open science leaders where we discuss how to fix academia. Easy, right? So hit that subscribe button, leave a rating, or find us on Twitter at MotionPod. But for now, let's get into the show. And this week we have Hannes Meyerborn to talk about the Exocyst, not the Exorcist, although to be honest, kind of feels like I'm saying the same thing. Okay, let's get started. Let's do it.
I mean, I'm basically down the first bottle of wine, so it's a good way to start.
I think we should leave that in and start actually.
I think that's where we should start. Yes, so this episode is going to be slightly unprofessional anyway.
I gave a talk on the member trafficking thing, and they asked me the same thing. Do you have a fun fact? And for them, I gave the fun fact that I spent both Christmas Eve and New Year's Eve on a plane after my PhD because I was being so done with everything that I just wanted to time out at the beach. Just gonna fly to Bali and spend a week on the beach and fly home again. And then I started my postdoc. So I think that is a fun fact that can be used.
There you go, John. If you need a fun fact, there it is. If you need one. So if you've just joined us on that note, today we're speaking with someone who we know quite well from the show. So I think everyone is aware that the three people involved in making the show are all friends. But amazingly, we have friends outside of the show. One of those is Hannes, who is just downing his millionth glass of wine now. And Hannes is a postdoc up in Dundee, and he's just posted an interesting preprint that has some really cool microscopy images associated with it. And I know I keep doing this. I keep bringing up really cool preprints that have amazing images, and this is not the format for images, it turns out. Somebody could have told me that before we started a podcast. But we might get lucky. Hannes might share it again on Twitter and see if we can get some more likes and get his serotonin levels even higher than they are.
Yeah, if you go to the Twitter account after the release of this, I'm sure we'll be able to get you across some of the pretty pictures. And they are worth looking at because they are very beautiful, some of them. If you like coloured spheres.
Yeah. And in case you haven't been able to tell, John's going to be joining in a lot more on this one. Emma, unfortunately, isn't here, but is sad to not be here and sends her commiserations.
So I'm Emma. You can't see it, but I've got a wig on.
So, thank you for joining us, Hannes.
Glad to be here.
That was nice. We like having you here. You don't have to like being here. Okay, so your preprint is all about the Exocyst. And you kind of got to be careful how you pronounce that, because if you get that wrong, people are going to think you're doing a whole other completely different kind of thing.
Yeah, it is a scary exorcist. It's a big, scary complex. Definitely true.
So, could you tell us what an exorcist is and why it's important?
Yeah, definitely. Well, the name is a bit of a fun one because if the scientists are listening, they know that scientists also like to have fun every now and then. And if people from the Drosophila field are listening, then they know they have even more fun with naming things in very bizarre ways, quite often just to make fun of people who try to pronounce it. And I think similar things happened with the Exocyst complex because it is very similar with the Exorcist, obviously. But the Exocyst is a complex that is crucial for exocytosis. That's where the exo comes from. So this complex is an octameric complex, octa meaning 8. It is quite large and is responsible for the tethering of vesicles to the plasma membrane during many steps of cell trafficking, but mainly doing polarized secretion. So we have to think about cells in different polarized states. If you have a migrating cell, for example, it'll have a leading edge. And if you have an epithelial cell, it will have cell-cell junctions. And if you have a neuronal synapse, it'll have neuronal synapses, right? And all of these polarized structures are made up of vastly different proteins, but they all need to get there in the first place. And the protein complex that targets the vesicles containing the proteins that make up these structures to the plasma membrane is this Exocyst complex. And that's what we have been working on.
And we've already mentioned your pretty microscopy movies, but you essentially end up with these images of these really pretty looking circles. Is the best way I can use of describing it. Could you explain what they are? So your model for how you study this.
Yeah, definitely. So I would invite all of the viewers, obviously, to hopefully look at the
Listeners,
preprints.
Hannes.
Listeners, no one is viewing me. That's good. Well, so I invite all of the listeners to definitely take a look at the preprint and take a look at my Twitter. My Twitter is Hannes, my name. And there I have a big thread where you can see all of the Pretty Circle movies in action. Because sadly, in the preprint itself, we did not upload any of the movies because we just quantified the movies. So you just have stills and graphs and numbers. So it doesn't look as impressive as it does if you upload a GIF or a GIF, wherever you are from in that debate. But yeah.
Let's not start that stall.
Basically, those Pretty Circle movies have been very successful for me on Twitter. And because they look stunningly beautiful, but what they are is obviously not just pretty images, but a scientific ways to measure the interaction of the exocyst complex with membranes. So these circles are actually spheres, and they are 10 micrometer large silica spheres that are coated with a lipid bilayer. Then I image them with confocal microscopy, meaning we take one section throughout those beads, and therefore they look like a circle, but they're actually spheres. And these spheres are coated with a lipid bilayer, just like the cell is surrounded by a lipid bilayer, right? The cell membrane. And these are so-called supported lipid bilayers, and they are a very good model system to probe in the interaction of proteins with the membranes. And then by having fluorescent tags at the membranes and also at the protein, we can visualize this whole system in a dynamic manner with the microscope.
And so you've not just visualized it though, what you've done is you've characterized how this exocyst sort of comes together and forms. So the exocyst is made up of two big primary subunits, right? So how have you managed to do that? How have you been isolating out these different components?
All right, very good. So we can't go into very much detail of the biochemistry, and I don't know what the listeners are.
I'm keen, but I don't think the listeners will be.
I do think biochemistry is fundamental to all life. So I think kinetic rates, biochemical interactions, specific binding, all of these are fundamental to all aspects of life. But it obviously depends what level of research people are involved in. Some people nerd out heavily about a binding curve, and some people are just like, whatever, it binds, it makes it complex.
You've just described me and John pretty well there. John's the massive nerd, and I couldn't care less.
Exactly. So I would put it brief. What we've done is we have all 8 separate subunits of the octamer in viral backbones. So from these plasmids, we then produce baculovirus. So these are viruses that infect cells, but are not harmful for humans, for example. So from these viruses, then we infect insect cells. So insect cells we use, so they are SF9 cells from like some stink bug or whatever, I don't know. Like John, I'm sure you know the name.
Sporido
fungu
or something. Yeah. No, it's not a stink bug. It's we'll fact check. I can never pronounce it. I know it when I see it, but
Spodoptera frugi. You know what? SF9 cells.
That'll do.
Exactly. And we basically use them because they are proper eukaryotic cells that are very easy to grow. They need very little to grow them in, but they divide and they produce proteins very well. And if protein biochemists are listening, then they will probably know that you can make proteins from bacteria or from insect cells or from proper mammalian cells. These are kind of like the 3 steps that you can go in. It depends how complicated your protein is, what expression system you want to use. So we are using the SF9 cells. And basically, to make this not too detailed, it's always difficult. I don't know what to say. We.
You did you physically did
we, exactly.
That's what we're all about on this podcast, promoting the people that actually did the work, you see.
Exactly.
Yeah.
So what I did in the first month of my of my postdoc, I'm sorry, not PhD anymore. In the first month of my postdoc, I've made all of these viruses of the 8 exocyst subunits with various different tags. So all of the 8 ones with 3 different tag variations. So 8 times 3 is 24. So I've made all of these different viruses. And then I started to assemble the whole octamer piece by piece. A bit like from Lego, where you add one brick to the next one, I started to assemble it from dimers, then to tetramers, pentamers, hexamers, heptamers, finally to octamere. So that was a decent amount of work. I've done probably 200 or 300 pull downs just to really know what subunit interacts with what subunit. So I expressed them separately and then did pull downs to like see what comes down with what. So that was a lot of the groundwork, was pretty hard grind work.
If only those were pull-ups, you'd be ripped.
Well, for every pull down, I do a pull up.
We do jokes too at PPIM.
So yeah, but I think like all of this grind work was quite important to really set the basis of how the whole octameric complex interacts with each other. And I think a lot of listeners are probably scientists know that in every project there's a time for just stupid grind, and sometimes it just has to be done.
We've all been there.
So with the, I just would, so obviously you pick sort of insect cells, which is kind of the middle ground, I guess. Is the reason why you couldn't do it in bacteria? Is it a folding issue, or are they subunits glycosylated, or what's kind of the why specifically? Why did it have to be eukaryotic rather than prokaryotic cells?
Yeah. So a lot of this has been, we do benefit from the body of scientific knowledge. So it has been tried in the bacteria and it was unsuccessful. And like one of the reasons why the human complex has not been purified so far before we came along is that it is not that easy to do from bacteria. So it is an octameric protein complex of total size about a megadalton, almost a bit less, and has a very complex folding pattern and is very foreign to bacteria. So bacteria do not have a very sophisticated membrane trafficking machinery because they're just like single cells, and they don't care very much. In eukaryotes, where we have multicellular structures, it is much more important to traffic the right thing to the right place. So a lot of things like GTPases and trafficking factors are not that well known to bacteria, and therefore they just like stick it in inclusion bodies or it aggregates. They don't know what to do with it.
Sorry, I was drinking at the wrong time there. I thought you were going to keep on talking.
Well, I can keep on talking, but I also want to give you some space to like ask questions. I don't want to be like in 1 hour monologue of Hannes talking to the camera, because that can happen easily.
So bring it back to general Exocyst stuff. One of the things we've not mentioned yet, but is a big part of the preprint, are pips.
Yes.
Which are phosphorinosatides. And I'm only going to say that once.
That's all right.
I'm not going to get that right again.
We can stick with pips.
Stick with pips. So pips are these things that kind of give membranes a bit of an identity, right? So they help signpost where the cargo is going to go, if I'm understanding things right. So have you tried switching out the pips and seeing what happens to the cargo? Does the cargo within the Exocyst still go to the right place? Does the Exocyst not form? Or does your cargo get lost somewhere? Or does it go to the wrong place?
Yeah. So the pips are very interesting, and I've gained a lot of appreciation for them during my postdoctoral work. And you mentioned correctly, they are a signpost for membrane identity. So what they are a group of phospholipids with specific head groups that are differently phosphorylated. And depending on where in the endolysosomal system you are, your organelles will have a different identity of phosphoinositodes. And through our work, we really highlight the importance of the continual interchange of the identity of membranes. As the membranes get trafficked to different places throughout the cell, they change their identity and they change their phosphoinositide code. So we could show that as a vesicle travels towards the plasma membrane, which has the identity of PI45P2, it gains this identity to go there. And I think that is one of the things that I find quite fascinating for my own work and also for my future work, is how this membrane identity is in constant flux and is being constantly changed by the activity of kinases and phosphatases that change these pips around.
So is this impacted by sort of the cell's environment, so things that are external to the cell that it might be taking in? So, for example, an immune cell, when it comes activated, will upregulate its glycolysis pathway, for example, and that has a whole bunch of knock-on effects that we are only really recently starting to understand what that does to a cell. But is this something that can come into play on this kind of thing? And because, I mean, presumably these enzymes are used elsewhere as well.
Yes, interesting question. There is a lot of fluidity within the PIP pathways, right? And in different stimulations will have different effects on cells. But I don't quite know. All right. So if we look at different stimulations, if we look at differently polarized cells, for example, then you have the phosphoinositides being localized at different places. For example, if you have cells that you stimulate with bacteria, they will engulf this bacteria in a process called macropinocytosis. And this will involve phosphoinositide 3,4,5 phosphate, so PIP3. Yeah? And that will be generated at the place where really these bacteria or spheres or whatever you give to them are being taken up. And then you also have different phosphoinositides in the lysosome and different phosphoinositides in the mitochondria. And with different stimuli, they will act differently. But it is still a bit of an area of research about like how they act in what stimulation. So for immune cells, I think it would be quite interesting to know.
Yeah, I mean, it kind of, your description of how it changes reminded me of a talk I saw years ago now by Judy Allen, and she was talking about how in neutrophil development the lipid content changes over time, and that is crucial to like normal development. It just kind of reminded me of that. And the other thing that sort of this has rounds for with immune cells is that immune cells, when they secrete things, it's very directional, right? People from their textbooks seem to think immune cells just secrete things at random directions, but it's not. If your immune system has a bacteria in front of it and it's going to secrete enzymes or proteases, whatever it's going to secrete out, it does that directed at the bacteria in front of it, not just out to the side somewhere useless. So it was just interesting potential.
I think like what is interesting me and what I've thought about doing in the future, if I ever gain independence, would be to look at the immunological synapse between a killer cell and a target cell.
That's what I'm thinking of, yeah.
There you have two membranes that are very close to each other. So they're like, what happens there is like the killer cell attacks the target cell and like really like latches onto it like a vampire and starts biting it and secreting it. And this zone where they form a contact is a very specialized zone. And the phosphoinositides in that zone are also very heavily regulated. And it is still a bit unclear about how these vesicles then fuse to that synapse and then release the toxic agent that kills the poor target.
You should chat with Dan Davies at Manchester. He's like the big name in immune synapse stuff and does a lot of really cool imaging stuff. Anyway, moving away from immunology. Go on, John. Ask us a question that isn't immunology.
No. Well, yeah, I was going to say. I think this was just again with the whole membrane identity thing. This was something I had such an appreciation before this. Obviously, I knew the membranes had these different identities, but yeah, just thinking about it. When a vesicle merges into a plasma membrane, you're like, well, surely that's going to end up as a little patch of like a different membrane identity, which is then what this is kind of all about. It's about like matching it up as it goes. So maybe this is a nice moment to talk about ARF6, which is this protein that's very important in this process. And I have a question on that in a minute, but I'll let you give a little bit of background about what ARF6 is first.
All right. Okay, so ARF6. So let's start at the beginning. What are GTPases? I do not think it is a trivial thing, and I do not think that most people know what a GTPase is. It's a fun side topic. We can talk about it at another point. Like my girlfriend is a non-scientist, so whenever I talk with her, I am very cautious to like back it up as far as you can. So ARF6 is a small GTPase. Small GTPases in membrane trafficking have a switch-like behavior. They can be in an on and in an off state, depending on if they have GDP or GTP bound. So this is guanosine triphosphate or diphosphate. And these two nucleotides can be exchanged by the function of exchange factors. So this gives the cell a mechanism to specifically turn on and turn off these small GTPases, and then effector proteins will bind specifically to the on state, sometimes to the off state as well, but mainly to the on state. So it is a question about how do cells regulate spatiotemporally, like where membrane goes. And this is done through these small GTPases. And ARF6 is one of these small GTPases. There is a whole family of them. I think in total there are, it's difficult to put this on because people will judge me if I say it wrong. There are hundreds of small GTPases out there.
Small.
Yeah, it's a very complex field, and there are a lot of them. And basically we could identify one of them that is specifically turned on. And when this ARF6 GTPase is being turned on, it recruits a lipid kinase called PIP5 kinase that phosphorylates these phosphoinositols and changes, therefore, the identity of the membrane. So this is a mechanism about how the cell can say, now we change the identity of the membrane. It switches ARF6 on, and this leads to a change of identity of membrane.
So my follow-up question to that was going to be about, because obviously R6 is kind of specific to that. Like, how does R6 come to be on one of these vesicles? Is it just vesicles coming from certain places, sort of have it pre-attached? One's coming from the kind of the Golgi.
Are you asking the $1 million question?
Is that the one we don't know? Okay, yeah, I didn't know if this was known or not. But...
No, like there's this big dark horse in the field of membrane trafficking that is like, how does the GTPases go to the right place? Because obviously you always try to like find the factor before, and you always try to step back, and you always try to find out how things go to where they need to go. And this is most of the time in membrane trafficking where the thing stops. Like the question, how do the GTPases in the first place go to the right vesicle? That is a question that is a big factor in the field. And there are hypotheses out there, and I think it is mainly stochastic. I think it is a bit of random, but it's also unclear. Yeah, because, well, it gets very complicated. I could spend just an hour about like how the GTPases go to the membranes, but it is very complicated.
Well, there's another question you can kind of speculate on then. So one of the things you show is that the two subcomplexes bind these coated beads stronger than the entire complex does, which would kind of seem a bit counterintuitive. So, I mean, why is that? Do you have any ideas why that might
All
happen?
right, yeah, sure. We're going into the details. Definitely. Yeah, definitely we have an idea.
Guess who wrote this question?
So I do hope we address this in the preprint. If not, then definitely I'm happy to clarify it. But it does get very much into biochemistry now, so listeners, be aware. If you don't like biochemistry, maybe put some fingers in your ear.
Don't leave us.
Just skip ahead like a minute.
So like in biochemistry, what is essential for life is binding kinetics. And everything has an on-rate and everything has an off-rate, and everything binds to something and unbinds to something. That is the truth. And that is the case for every interaction in biology. The only thing that doesn't unbind is if you have a covalent interaction where something are really covalently bonded, then they will stick to each other forever. But otherwise, you always have an on and off rate. And what we could show is that the two halves of this whole octamer complex bind to these lipids with very much the same affinity, meaning the same KD. KD doesn't tell anyone anything if they are not biochemists, but the KD basically is the
same strength.
Yeah, exactly. It is a measure of binding affinity or strength. Like the lower the KD, the higher the affinity, the higher the strength of binding is. And both halves of these complex bind to membranes with very much the same strength. But then if we put these two halves together, they bind with less affinity. And this was a big surprise for us. Because if you think about binding kinetics, you have to take into account affinity and avidity. Now, affinity is an overall measure about like how strong the binding between two things are. Avidity is now, if you have two things that bind to the same substrate, their separate affinity should add up to make the whole avidity higher. So you can imagine it if you have two parts that bind to the same thing, then the separate affinity of these two parts should add up to give you a higher avidity. And I know this is very complicated for any listener, and I do not know if we should really talk about this in a podcast because it is pretty hard biochemistry.
I asked this question because John said you had a good ship anchor analogy.
Yes.
I've not heard this yet.
We do have a ship anchor.
You went straight science.
We've not got to that analogy yet. It's coming.
Exactly. So we don't see an avidity effect between these two subcomplexes to the membrane. Therefore, we argue, and we can show that only one of those binding interfaces binds to the membrane at once. So it is situated in trans for the biochemists. It is not in cis, where it is on the same side, but these two binding interfaces are in trans, which are on the opposite sides. So if one side binds, the other side cannot. Because if you think about it, one side could be on the tummy, one side could be on the back. You can't bind to things at the same time.
Is the other side binding anything at all?
Yes, definitely.
Yeah, that's how it links the vesicles.
Yeah, exactly. Definitely. So this other side is then able to bind other membranes. So this is how it tethers two membranes to each other, we think. That it is able to have two binding sides, but they don't bind to the same membrane, they bind to opposite membranes. And now the question is, why is the affinity of the whole complex lower than for its halves? That one is not easy to answer. But the important point is it is not higher. If it would be higher, so like a lower KD, then we would know that there was an avidity effect between two binding interfaces binding to the same membrane. But since it is lower, you can say that it is not the case. And then there's an analogy that I like to use to visualize it in my tiny brain is if you have an anchor that is trying to anchor down a ship, and you have the two half of the complexes have the same anchor, but they only have to anchor down half of the whole complex. So it's much less to anchor down. But now for the whole complex, you have to anchor down something that is twice the size. So it is like comparing anchoring down like a big sail ship to like a massive cruise ship. Because even though you have the same binding interfaces, they are only present in half of the protein or in the whole octamer complex. And I think that's like how I try to make it work in my head.
So I think that makes sense. If you use the same pathetic teeny tiny anchor from a sailboat on a cruise ship, the cruise ship's going to just plow ahead and keep going.
Exactly. And that's why we think that there's a less affinity of the whole complex to the membrane than of its separate parts, which are smaller. Yeah.
Are the two parts the same size as each other, or is one part slightly bigger?
Pretty much 50-50. Yeah, they're pretty much. I think like one is 350, the other one is 400. So yeah, they're pretty much the same size. And this is all an ancient duplication event of evolution, like happens so often.
So has anybody tried knocking down or out or all or some of the Exocyst components? And if we do that, what happens? I mean, presumably if you miss one of those parts of each side of that thing, you don't get your whole Exocyst form, and then it can't do its job,
yeah,
right?
Definitely. So it has been done by a few people, and there's also some very exciting work coming out soon from a PI in Cambridge, who I don't know if I can name it on here publicly, but he has done a lot of these knockouts. And basically what has been seen in the field is if you knock down or out any of the single subunits, you get a drastic decrease in vesicles that are being tethered to the plasma membrane. So all of the 8 subunits are important. If you knock down or out any one of them, you get less tethering to the plasma membrane. And for people who are interested in mice and development, I think it is incompatible with life.
Yeah, I was going to say that sounds like it's going to be, if you try it in vivo, in vivo in animals as opposed to in cells, that's going to be embryonic lethal by the sounds of it. But
yeah, like even cells don't survive long. Like even cells in the culture dish don't survive long if you knock them out.
Really does look like I've been stabbed in the shoulder.
Exactly, that's what I thought. Like, oh my god, it happened.
Johnny's just had a new tattoo coloured in today. For anyone that, if I leave that bit in, that's what that was. He's in a bit of pain. He keeps wincing.
So that's all the questions I had about the preprint. I think we've covered it pretty well, actually. And I mean, I knew this anyway, but I'm going to say it in front of people for a change. But you are very good at explaining complex things.
Yeah, yes. It is because I live with the non-scientists at home, That is because why, I think.
I did. I do have to refer people back to your original Twitter thread with all the images, which has got actual, the sort of scientific, kind of graphs and the proper figures and things. I suppose just kind of schematics, but it is nicely explained, and I do recommend people to go and have a bit of a look at it.
God, we're plugging your Twitter account so much.
Yeah.
Although to be honest, you've probably got more followers than our Twitter account.
I do.
So some of Hannes's followers would like to come over to us.
It's a pretty circle.
We'd greatly appreciate that.
It's a pretty circle. Twitter is a fickle beast, isn't it? It's very difficult to play the Twitter game. It's not easy. You have to have the right amount between pretty images and science and non-science, and I don't know. It's very difficult. I don't really understand.
Yeah, you can't just do what I do and vent.
No, you shouldn't.
What's really interesting is that I know all of the guests we have on seem to follow Emma and John on Twitter after the shows. None of them follow me. Some of them do now. But for quite a while, there was this habit where they would all follow John and Emma and not me, as if they hadn't just spent an hour talking to me.
That was enough.
Maybe they checked your Twitter account and were like, no.
I know, I probably do.
Where do I find out about the different bioarchive licenses? This CC, BY, CDXY nonsense is driving me nuts.
ASAP Bio have a resource for that.
Ugh, that's your answer for everything.
That's because they have everything you need to know about preprints.
Sure, they probably have the basics, like info on the preprint servers, but what else is there?
There's so much more. Looking to post a preprint, but not sure what different journal policies are? They have a collection to help you out with that. There are meetings around preprints and associated services. If you want to know how preprint adoption has changed over time, there's even a page on that.
And COVID?
They have a big section on preprints and the pandemic. plus some really cool infographics for communicating preprints.
And university policies? Surely they don't have that.
They collect uni policies where possible.
Okay, they do sound pretty impressive, but is it not a bit of an echo chamber?
It can be, but ASAP Bio also engage with people who don't love preprints and have concerns. So we had an excellent discussion on this very topic a couple of months ago.
Oh, is there anything ASAP Bio don't do?
Honestly, no, they're so nice over there. They were so quick to jump in and support this show. It's your one-stop shop for info on preprints and open science initiatives, so head over to asapbio.org to learn more and subscribe to their newsletter for the latest in preprint news. If you want a deeper dive into the world of preprints, then look out for the next recruitment of ASAP Bio Fellows. So, one of the things we like to do on the show, I'm not sure if Hannes listens or not. I assume you've listened to
Don't
some.
pull that thread.
Don't
Don't pull
answer that
that thread.
question.
Don't pull that thread.
I do sometimes.
So one of the things we like to do is talk about not just the preprint itself, but the process of preprinting, publishing fields, and just your experience in academia. So that's this section of the show, now that we're back from our little break. So we have the benefit of knowing you, so we kind of know some of these answers anyway. But I think compared to the other people we've had on, your journey towards why this has been preprinted is slightly different, I think, so far, if I'm remembering correctly. So where did the decision to preprint this work come from?
So for me, it was very important to get this pre-printed so I can apply for my own independent fellowships. So I made the point quite early on with my supervisor that it is important to have something out there. Yeah. For everyone who's a scientist listening who knows, if you put on your CV, submit it to Nature, well, everyone will laugh at you, right? Because you can submit everything to Nature. It doesn't matter. It doesn't mean jack. Yeah. But if you put on your CV this paper, a link to bioRxiv, that is usable. So then the funders or the agencies or whoever you apply to, whoever you send your CV to, they can actually click this link, open it up, and see what you've done. And this was very crucial for me. So because there are a lot of funding agencies and funding schemes out there, and a lot of them are very much focused on early career research. And it can take quite a long time to get things genuinely published in like a real journal. So to put out a preprint gives us early stage career researchers the chance to show that we have done our work and then apply to fellowships or grants with this on our CV, because it can take at least half a year from submitting something to actually publishing it. So it is a long and strenuous journey, and half a year is a lot of time when we are talking about grant submissions and fellowship submissions, and it is quite important for that.
Half a year is one application cycle.
Yeah.
So if you're waiting on your paper to come out, you've missed your first chance.
Yeah.
So you haven't just preprinted this immediately though. So you did send this around journals first. And I think one of the things that has been an issue is that this work straddles two different fields. And so it is a bit difficult. I think whenever work does that, it's always difficult to find the right home for it, because non-specialist journals don't want it because it's not non-specialized enough. Specialized journals don't want it because it's not specialized enough, and you can't really win. So how's that process been?
Yeah, so this whole process,
other than horrifically stressful,
this whole process is still ongoing. So there is not an endpoint towards this yet. And we can ask ourselves why. And that is a very big field of debate. So why do people have such a hard time publishing things? Even though we have gotten a tremendous good feedback. And it's always, for me, very puzzling. You present this work on like conferences to people in the field who know their stuff, who really do know their audience. And you get like good feedback. Everyone is like, oh yeah, that's great. I'm sure it's going to be fine. And then you submit it to a journal and the editor says like, nah, it's like, I don't know, like, where does it come from? Like, I don't know. Like, do editors have too much power that they can just say, I don't like it? It's like, I don't know.
Yeah.
Like, and they don't even need to give you a reason. They can just say like, nah, nope. Like, you're done. I don't know.
It seems like we're in this strange situation where a lot of like journal editors are sort of full-time journal members of staff, and so they haven't done science themselves for many years, whereas the reviewers are actually usually active scientists. So it's this sort of strange thing of people who are actually kind of out of the field having that initial gatekeeping opportunity on something they haven't generally done for
and you can't
a long
even
time.
argue with them. That's the worst part. Like you send it to the editors and they say nope, and you just have to like say, all right, we go on. Like when you get reviews and they say we don't like your work, you can at least argue back and say, we have done this, we've done this. I think you're wrong because of this reason and that reason and this reason. But the editors, they can just say no, and you're done. You're out. And they don't even have to say why.
I mean, we often bang on about the public peer review, about how kind of the peer reviewing kind of comments from paper should probably be kind of attached to and published alongside the paper, and they're not always. And especially that kind of decision would be very useful to have as well.
So does having a preprint out make you feel better about that whole process? Because, like you said, you've had a lot of good feedback. People have you were throwing numbers at us before we started the show properly. So maybe you want to do that again. But the paper has had a lot of attention. So it's clearly something people in the field are enjoying.
Yes, I think that putting up a preprint can empower the scientist and the individual in a way that without it is very difficult. So from this preprint, just in the first 2 or 3 weeks, we've had like 500 downloads, thousands clicks and things. And it does make a difference towards me feeling appreciated, and like me feeling that my work is actually being read and being seen. Because we've also submitted our work to 3 journals so far, where it has all been desk rejected. And that would make you feel like, oh my God, my work is rubbish. Having this dichotomy between the actual readership, the public, and the editors points out to me that there is something different. There's something going on where the actual scientific community really likes the paper and actually thinks it is pretty fire, while the editors are like, nope, don't care about it. And I don't quite know where that comes from.
But of course, now that you've done that, when you next send it to a journal, in the cover letter, you can say, well, we've had this many downloads, this many people have read the thing. And then you can show that actually the community are interested, and maybe that might sway editors. I do that with my past few papers. I don't know if it made any difference or not, but I know other people who also do that. And, they say it does help to get past that initial hurdle. Maybe if an editor's not familiar with that stuff, it makes them maybe think twice.
I mean, I think at the basis of this all is a very complex system with no easy answers.
We get that answer a lot.
It is a simple truth that there are way more papers being put out than can be put in any journal. So there does need to be a triage somewhere where someone at some point has the power to decide what is hot or not. And that will always lead to hurt feelings. And that is just a point about our scientific system that we're in. That there is, if we think about capitalism, there's a demand and supply, right? And our world is built on a capitalistic basis, as much as I disagree with it, about too much supply of papers to too little demand of journals editors. So there's a power imbalance where the journal editors can very much sit in their big chair and say like, nope, yes, nope, so they're like Caesar being like giving the thumbs up and the thumbs down about like
their throne of skulls.
Yeah.
This is curation process, I suppose, is the word we're looking for.
That was one of my questions.
Well, no, curation, I think it's more than that, and I want to be honest with everyone listening. I do think there is a case for high impact journals and low impact journals and a triage of it, because I know, and every scientist does know, how much there is published every year and how much one can actually read. When we published our work and I looked at the alt metrics, like whatever that is good for, we were at like the top 5% quantile, out of 20 million research outputs, we were place 100,117 or whatever, the number. But that just makes you think like there are 20 million research outputs tracked in the same time. It's like, good Lord, like who can like read 20 million research outputs? And like, how do you sift through all of this research in a way that makes it visible to people? Because that is my main point. It's like I've spent these last 3 years busting my ass off, working really hard, and I do not want my work just to be lost in the cloud of 20 million papers.
A dusty cupboard.
So I mean, this is something we occasionally talk about and get into. We'll not do it here. But, you say there's a place for high impact and low impact, which, okay, we'll take that. That's fine. But what do you mean by that? Are you meaning high impact as they currently are, which is very general? Or are you meaning that we need a definition within the field? Because, we've got all this science being published, and a lot of it is a bit pointless. And I think the question is not where it's published or how it's published. It's more the basis of what we're actually asking and doing. I think we need to go right back to the start of the whole thing to really sort that problem out. But are you arguing for general journals or arguing for specialized journals? Because there, I think there is a difference.
Yes, definitely. So I think I, for myself, differentiate very much between what is in my field and what is not in my field. I will read everything that's published, wherever it is being published, that has to do with what I'm working on. I have a PubMed alert. I look through bioRxiv with my hit words, just like the Exocyst and Clathrin. That's what I used to work on. And I will read it, no matter what the journal is. And I will read at least the abstract to get an idea of it if I think it's interesting. And I don't care where. So that's that, And then there is all of the research that goes on outside of my field that can be very impactful, but I would never be aware of if it would not be published in this journal that gives it visibility to me from outside of the field. So these are two very different things that need to be distinguished, right? So I would never look at journals that are very out of my field and then, but if something is being published in Cell, Nature, or Science that I never had to do with, but it's like really a great, profound finding, and I go on these websites every now and then and I see it and I think like, oh wow, that's really great, that's really cool, that's what they're for, and that's, I think, fine. I don't mind that.
I think it's what they should be for. I think at the moment a lot of what goes on in there is just, have you done some single cell sequencing? Oh yes, great, we'll publish you.
Have you got the words phase separation in there?
Yeah, exactly. That's coming from an immunology perspective. Anything immunology at the moment that is single cell sequencing and maybe one other hot technique kind of goes in those, and it's not the most cutting edge science, a lot of it, from, I can say that. But I mean, if you were coming new from the field and you didn't know that, you would assume this is all amazing science, and it honestly isn't.
Yeah.
So I understand your point.
You're definitely right. There are a lot of problems with it, and it is not, how it is right now is not how it should be. But I think in general, there is a point in my mind to have like selected journals that are really there for like the high impact science that everyone should read, no matter what field you're in.
Yeah, I mean, I wouldn't completely disagree with that. The problem, of course, comes is that the people who get into those journals will still use them the way they do now, which is, well, my work must be better than everyone else's work, and therefore I deserve that professorship, and to bully and abuse my staff however I like, as some of them have been doing. How do you get around that?
No one is arguing for bullying and abuse, and like obviously no one will think that. But we are also in a reality where we have a capitalistic system where there is a lot more supply than demand. So how do you sit on a panel and select if there are 50 people out there who are all good scientists and all want the same position? How do you select the one for the grant position? Right, and that is the question that we need to ask ourselves.
See, my argument would always be that you should look beyond the science and you should look at what they do to make academia better, what they do for outreach or teaching activities.
Like my job is science. I want to be judged as a scientist. I don't want to be judged for things that I do outside of my job. You don't judge me if I help an old lady across the street.
No. Okay, yeah, but your job as a postdoc is to do science, but your job as a PI is not just to do science, it's to teach, maybe, or it's to mentor students, or it's to do outreach and educate the public. Educating the public, I think, is a fundamental job of being a scientist, whatever level you're at. Because if we don't tell people what we've done, is there any point doing it, really?
Well, I would argue against that. I think that there's a very different skill set to being a good public outreach person than to being a good scientist. And I think there's a big fault within the hiring process where scientists are being hired on the basis of like high publications and like high scientific merit, but they actually are not good teachers, they're not public outreachers, but yet we all expect them to do it. Like, so we expect someone to be able to do everything only because they can publish good and do good science. So what we should do is we should separate this from each other. We should have dedicated outreach office that actually does outreach. We should have dedicated teaching people that actually love and want to do teaching. We should have dedicated scientists who love and do science. But like, to expect all of it from one person is just stupid.
It is, and I fully agree. But then at the same time, then you've got a situation where, what if somebody likes doing research, but because they've spent time, or maybe it's not even choice, right? A lot of this comes down to the fact that often it's women who do these kind of extra things, right? And they get the higher workload. And we're not including childcare stuff and anything outside of work. We're just including things that do work. So they sit on more panels, they teach more, they do more of the general stuff that nobody ever really accounts for, but is essential to a department running well. What if they want to be someone who primarily does research? They can't compete with those other people because their time is being taken away from the research time. And it's not always a choice. Not everyone can say no to the big boss who says, you're going to do this for me. Or, even will you do this for me? It's not always a question. Sometimes it's you're doing it. I'm just going to pretend to ask nicely. So, I mean, that's where I'm getting at with we should account for other things. But maybe we do need, I think we need to rethink the hiring process is my point. And it shouldn't just be you do really good science that comes in all these high impact papers.
I agree. I think we need to rethink what the universities really want. Do they want to hire a good scientist? Do they want to hire a good teacher? Do they want to hire a good PR person? These are three different things. And I think it is unrealistic to expect one single person to be excellent in all of it. So why don't you just split it apart, do 3 different jobs for 3 different people? That would be my suggestion.
I mean, so certainly from the UK perspective, some universities are now kind of doing that. So we have teaching fellows, for example, whose the whole point is you're independent, but you teach. And that is your career track. It's the same track as the traditional sort of research track, but you're just teaching instead of doing research. So there are, I guess, attempts.
And for me, on a personal level, I find it immensely stressful to have this obligation of public outreach and all these things pushed onto me because I need it now to get my grants, to get my PI, to get my independence. It is something that is enforced onto me, even though it is not what I am good at. It is not what I really want. I'm a good scientist. I want to do science and I want to get funded for my science. And I know there are a lot of people who are very good at public outreach, who love doing it. And why does it need to be me? Why do I need to be able to do everything? I want to really do good science to help push forward, the scientific community. But I don't really want to do all of these outreach things. Sorry, I'm honest about it. It's not really what makes me happy.
I don't think there's anyone who would disagree with you that we get pushed on to do, what the classic trope is, an academic is doing the full-time work of 7 to 12 other people's jobs, right? It's no academic is out there just doing one full-time job. They're all doing multiple jobs that they shouldn't be doing. And there are very, very few people, if anyone, who can confidently say they can do all those jobs well, because it's not possible. And the skill sets are so different.
You're very right. And even in my PhD, it was forced onto us to have one outreach yearly done, So you would go to some science fair, do some things. And that's fine. I think these are very, very important and crucial things for science and public outreach. They're very important to do. But I don't think you can force it onto the people who are actually not wanting to do it,
One of the other things we can talk about is being a postdoc, being the first person really in a new lab. So this is something you've just done as your postdoc. It's something I seem to keep doing for some bizarre reason. So, I mean, how have you found that experience? Because you came from a PhD in a lab with someone who, I mean, there wasn't a lot of people in the lab, but it was a very established lab, to one where essentially you're helping set the lab up from scratch.
Yeah, definitely. I think it's a good question, and I think it's a good thing to discuss for everyone out there who is on the verge of going from PhD to postdoc. It's a very difficult decision, and there are a lot of things to consider. And for me, I did my PhD, as you said, in the lab of an old established professor who was wonderful and I love very much, and I had a good time. And at the end, I was fortunate to have published a paper in a very decent journal, even before going out to looking for postdoc positions. So that put me in a very comfortable place to look for things. And I had many, well, we can do the long story, which is this. So the long story is, even during my PhD, I was always very keen about what do I want to do in my postdoc and things. And I reached out very early on, long before I finished, to a guy in the NIH in Washington. And I said hey, you're doing pretty cool science, and I think we could do something really cool together. Here's my paper. Why don't we have a chat and just talk about it? And he came back to me and we met on the phone at that time, because that was before Zoom was a thing. So we had like a long phone call and like really discussed everything we wanted to do. And then he said like, hey, yeah, why don't you come over to Washington? I invite you to come over and like we meet and give a talk and we find out what's happening. So I flew over to Washington, D.C., obviously long flight, and gave my presentation, gave my science, met everyone, and like all nice and good and very exciting. And it was very exciting. And I was really, really thrilled and I was really chuffed. And I was really like wanting to do it. And I said like, yeah, let's do it. And we would really like to do the postdoc with you. And he's like, yeah, sure. And we didn't need to think about it. You know, you go home and I will let you know what's happening, but it's very positive. You know, I think everyone really liked you and it's going to be fine. And so I was like, yes, it's going to be fine. You know, I'm going to Washington. Like, yes, ace. And then he was like, yeah, I'm just attending this conference. I'm going to let you know what's happening afterwards. Just going to need to speak to a few people. And then like a few weeks later, he was like, nope. You know, he said like, no, sorry, it didn't work out for some bullshit reason, you know. Sorry for saying it like that, but like honestly for some bullshit reason. That was not even genuine. So everything fell through. So then I was at the point where I had to decide, what do I do now? So I just went on jobs.c.uk and looked at like whatever postdoc positions were out there, you know. Yeah, so I had this whole big plan about like what I want to do with like for my postdoc, and like, you know, I was banking everything on it, and it just all fell through. So I had to look what else is out there, and I had to pull on my other personal resources. And then in the end, I had a decision between going to like a very big lab of like a PI that is publishing in Cell, Nature Science all the time, you know, with like 20 postdocs and like big money and all these things. And I thought like, you know, you have all of these postdocs and you have all of these people are working and you get like maybe one big paper out a year, you know. If you do the math, that means like that most of the postdocs don't get anything published, you know, that most of these people are just, you know, forgotten.
Yeah, just kind of middle authors.
There'll
on
be middle
everything.
authors, right? Yeah, you're not getting that one first author you really need out.
So then I also interviewed with my PI now, and he was a new PI, well, still is a new PI, on a Welcomeship Fellowship, Henry Dale, just setting up his lab. And he was still very much needing to succeed, right? So you have now two big questions to ask yourself. Do you want to go to like a big shot PI, a big shot professor who has everything he needs, who's like fine, he'll publish whenever he publishes, or do you want to go to like someone who's hungry and like needs to push and needs to succeed and wants you to get there? That's what I did. You know, so I decided to go with the new PI because I knew that just him and me, I would get proper training for in the first part, and his success is dependent on my success, right? So we are interdependent on each other. So therefore, he will make sure that he really wants me to succeed. That can lead to big problems or big success, you know, because the pressure is definitely there, you know, because you need to succeed in order for the lab to succeed. If you don't succeed, the lab won't succeed, and it's going to be disaster, you know. But if you do succeed, it's going to be even the sweeter because you are really a good team, you know. And that's what happened to us. We really went and we're a good team. Like we came along very well. We had the same ideas, the same vision for like where we wanted to take the project, and we really did do it together as a team. And in that regard, I got lucky, I think, because it can also be disaster.
Well, I could tell you the other side of joining a new lab where it doesn't go so well, which we've never done on this yet, not properly. I don't know if Emma would leave it in. I think Emma might cut it out.
I think Emma would suggest cutting it out. You'd probably suggest cutting it out in the cold light of day.
Well, it's fair to say that you had bad past experiences. There's nothing wrong with that.
So yeah, I mean that is, you know, that's probably a really good story of how to make that decision. Actually, there are a lot of benefits to joining a big lab in that everything's set up so you can kind of just get started. The other big benefit that you didn't mention here is that when you, I mean, in science, there's the expectation generally that you move, and generally when you move, you're moving somewhere you don't know anyone. If you join a big lab, you've kind of got a community, sometimes, not always, but ready-made for you because there's a lot of people you can go out and talk to and do social stuff with, if the lab's social. The downside of joining a lab on your own is that you're on your own, and you've got to hope that either the PI has managed to sort of integrate with maybe some of the new groups, or just another big group there, so that you've got that ready for you there with other groups, or you've got to do it yourself. And I think that's something that people don't think about, but should think about.
Yeah, you're right.
It's one of the things I found the hardest each move.
Yeah, right. It was a bit of a pity when I started in the lab and there was no one there but my PI. It was just the two of us. And then you have to reach out to other labs or to oh, dare I say it, Tinder.
And how is Lisa?
You can spend a lot of your time not just socially alone, but at work you can spend a lot of time alone. Like a lot of my work is microscopy based, so I'm in the microscope room all day long on my own. I can genuinely go to work for a week and not talk to a single person at work or not at work. So there are, I mean, if you're on the microscope, you're going to do that anyway. But until we moved back to the main office relatively recently, that was my week pretty much. There are difficulties.
Yeah, I agree. And it is more difficult once you get to the postdoc stage. I mean, for the listeners, I've done my PhD with both Johnny and John and Emma. So it was a time where we are really all in the same place, in the same circumstance, facing the same challenges. And that is something that ties one together in a way that is a very strong connection. And as a postdoc...
Well, we're still here.
Exactly, we're still here many years later. And as a postdoc, this connection is missing, because you are not in the same boat as the PhD students. You are not in the same situation as them. You don't have to get your PhD. You have a different challenge ahead. You have to get paper. You have to get this done. I found it very difficult to realize that we are not on the same eye level. It still hurts me a bit because in my heart we are still very much the same, but like not really. And like it does hurt me a bit when like the PhDs are now like going out. They're going to the pub and they don't ask me out anymore. Right?
I think PhDs are kind of, you've got a common goal. All you're trying to do is get your PhD. Whereas postdocs it's a lot more complex than that because we don't start together, but we're kind of in competition with each other now. So when you work with people, you work with them, but also there are unfortunately a lot of postdocs who will do whatever they can to get themselves ahead and don't want to work with anyone else. They will make the most of the opportunities presented to them, but they won't present opportunities to other people. They won't help other people particularly. I think anyone listening will have at least a few examples of people they know who are like that, because it is unfortunately common. Again, lots of very nice people who will help everyone. But I think postdocs are in a much more volatile position. And then you've got the added difficulty of the pressure to get a paper out. And so you have then you've got those postdocs who never leave the lab. So they might be the loveliest people ever, but you're never going to socialize because there is still in the lab at like 9 o'clock on a Friday night. And I mean, they shouldn't be. But...
And postdoc is a very broad term because you also get very different postdocs who are very much established people in their own right, who have a family, who have a very big life outside of the lab, who are mainly more middle-aged, more accomplished, and say, it is my 9-to-5 job. I go home, I have my family, right? But that is where I saw the biggest problem. I'm neither of them when I started. Right? I don't have my set family to go home to, yet I do not have the PhD student friends to drink with after work. Mm-hmm. So then I'm alone. So that was a big realization for me to come into my first postdoc, to like see what is my life and my point as a postdoc was quite hard. But then for everyone listening who cares, I found a wonderful girlfriend, and we are now very happy into the countryside, and it is all good.
Who I should say has been plugged at least 3 or 4 times so far tonight.
Yeah.
We're gonna have to add her in like some acknowledgement at the end now.
Thanks to Lisa, who deals with Hannah's on all our behalves. And you'll always have us as well.
Oh yes, I think that has been that's been the thing I've struggled with the most being a postdoc is that weird isolation, and you're in this weird in-between place where you don't quite belong anywhere, and it's difficult. Anyway, that wasn't a positive note to end on.
I was going to say I had a nice little positive sum up there, and then you brought it down.
Sorry,
it's all right. I think we'll leave all this in. I think it just it's this is what it's like behind the scenes, folks.
It's a mess.
Please stay tuned after the outro for information on a special event for EU and UK postdocs wanting to set themselves up as independent PIs. Unfortunately for us, but happily for you, it's contained within part of the bloopers section, so please don't judge us too much for that.
Okay, and that is the show. If you enjoyed listening, then hit that subscribe button for more and leave us a review on whatever platform it is you're listening on. You can reach out to us on Twitter at MotionPod or online at preprintsandmotion.com. Didn't enjoy that? Well, we're all scientists here, so send us your review and let us know what works or what you'd like to hear more of, or less of. But until next time, have a good week. Go to the toilet. Well, in the meantime, listeners, I have to keep you all entertained as the two old men leave us to go to the toilet to relieve their full bladders. I'm not really sure what to talk about. I could just insult them both, but that seems a little bit mean. Have I got anything to plug? No, I got nothing to plug either. Oh, I do have something to plug. Hey, here we go. It's always weird talking to yourself. It's I appreciate actors a lot more these days. So, a lot of people probably don't know this, but one of the many things that I've done when I'm not in the lab, which if you know me would sound like I'm not in the lab very often, but I actually am a lot. But one thing I've done is a few years ago I set up the UK and EU postdoc Slack group. And this is a group for any postdoc, well, any PhD student about to be a postdoc or anybody about to become a postdoc from other routes who are moving to the UK or the EU who are wanting to just be a postdoc and see what life's like and network with other postdocs. So as part of that, on the 15th of December, we've got a Pathways to Independence workshop, which is going to, in the morning, that will have a bunch of different PIs who've used different routes to get into their independence, and they're going to give a sort of 15, 20-minute talk on what it is they did, so their sort of career path from PhD to independence, and any tips they've got to offer on their particular pathway. And then after they've done that, there's going to be an hour-long Q&A session. It's being run through the Slack group, but also with UKRI and their Early Career Researcher Forum. So that we'll have a link to that, I guess, in the show notes. There you go. I filled the time by plugging my own shit.
Are you leaving that in then? Okay.
So, John, you are going to have to listen to that and move it to the end or the start.
Unlike some of us, I actually listened to all of this.
So, everyone is now back from their toilet break. This would be a really good time for the ASAP Bio ad to go in, I guess. We can all announce we're about to go pee, or the old men are about to announce they're going to go pee, and then we can do an advert.
Cut that bit out. We'll do the advert, and then we'll come back seamlessly. This is us coming back seamlessly after the advert.